So, to introduce yeast genetics, we have to talk a little bit about the yeast life cycle and the introduction to tetrad analysis. Now, what's critical for yeast is you can grow them in two states. You can grow them as haploids and diploids. So the basic gist is if you have a haploid line of yeast, Saccharomyces cerevisiae, you can sort of say 1N, right? And then these things will mitotically divide again and again and again. Right? Now you can actually do matings between haploid strains of yeast. So for example, you may have another strain that's 1N, right? And with a given genotype, and those things will divide on their own, either in liquid culture or in on plates. And you can mate them. Now, I'm not going to get into the genetics of the mating, but there's an important thing between the mating types, between A and alpha. But for now, let's just assume you can mate them, and then you can mate these haploid strains and make a 2N individual. Right? Now, that individual can also mitotically divide. Right? So you can also put that onto liquid culture, or you can put that onto plates, and you can do genetics with those ones. Right, so you can get a mitotically dividing group of 2N. So that's important. Now, if you want to understand meiosis, it helps to be able to convert these 2N strain via meiosis back into a haploid strain. And the way that you do that is through starvation. So if you take your 2N population and you starve it, you will induce meiosis. All right? And then from that meiosis, what you end up with, of course, is a tetrad, in which you end up with 4 haploid products inside a single tetrad. Now, with regard to genotyping, it's very similar to any sort of bacterial genotyping. We genotype oftentimes for the ability to produce an amino acid, for example. So, spore that you can grow up on complete media. This is a 1N spore that you've dissected out of a tetrad. You can grow it up on complete media, and it will grow fine as like a little colony. Here's your plate. You've taken your spore from a tetrad and you've dropped it right there. All right. Well, if you say, for example, put it on leucine minus media, or met minus media, you can tell what the genotype is with regard to the leu locus or the met locus. So for example, if this particular haploid spore that was dissected out and grown up on complete media is capable of growing here on leu minus media, then we would say then that one must be leu plus. Right? But if that same one then copied from this complete media to met minus media is unable to grow, right? So we would say that this particular spore that came out of that particular tetrad would be met minus. And that would basically be how you do your genotyping. So the basic genetics is that you do a meiotic, do meiosis, make tetrads, dissect out each of the spores and genotype each of the spores, and that way you can understand how meiosis proceeds for every single haploid product, as opposed to what we typically do with non-fungal types of analyses, in which you really only get to see one haploid product of meiosis.